I'm going to talk about epigenetics from my point of view. It's a huge topic. It's an equally huge topic as almost as trying to talk about genetics. So I'm only going to present my view of the elephant, so to speak. But certainly, when you consider what the word means, epigenetics, it refers in the sort of narrow sense, which is the sense that I'll take today, to the two main components of the epigenetic code, namely DNA methylation patterns and patterns of histone modifications in chromatin. And these are modifications of DNA and chromatin without any change in the DNA sequence, hence the word epigenetic. The patterns set up stable or metastable patterns of gene expression that are transmitted to daughter cells in somatic tissues. Now, the word somatic is important because these patterns are erased, in fact, for most genes, though not all, as we'll see, in the, well, actually, for all, let's just say, in the germline during gametogenesis. Epigenetics is a hyped topic, particularly in the last 5 or so years, since it became a roadmap item at the NIH, and there's been an explosion in NIH grants on epigenetics, and the conversation has become very much public, including somewhat of a mystique, which is unnecessary and has led to perhaps some misconceptions. And I'm being blunt about this. I don't want to criticize any of these articles or books. It's not the point. The point is that if you look at these identical twins, they're very similar, almost identical, in fact. And it's because they have the same DNA sequence. The truth is, Mendel and Darwin were correct. They remain correct. By and large, genetics calls the shots. And I'll have a lot, actually, more to say about this. There's been, a century of careful study on inbred animals, which shows unequivocally that acquired characteristics are not passed to future generations. There's no basis other than careerism for the sort of neo-Lamarckian pronouncements that one not infrequently sees today. But with that in mind, let's look at some genuine and important epigenetic phenomena and consider their mechanisms and implications for precision medicine or genomic medicine. From my point of view, here are the epigenetic truths. Modifications of DNA and chromatin are important for setting up stable and metastable patterns of gene expression in somatic tissues. These patterns are faithfully transmitted in somatic cells, but as I said, reversible in gametogenesis. Epigenetic phenomenon, the best studied of which are X-inactivation and genomic imprinting influence in utero growth and postnatal behavior. And aberrant epigenetic modifications that we refer to as epimutations can and do cause developmental disorders and cancers. And last but not least, there are widespread and important interactions between the human genome and epigenome, and these are important for understanding the pathogenesis of common diseases. So for those of you who don't think about epigenetics, I have to say that the first phenomenon that kind of highlighted many of the mechanisms that we know are important for all other epigenetic phenomena is X-inactivation. And X-inactivation in humans and mice, in mammals, is a way of solving this universal problem of equalizing gene dosage from sex chromosomes in animals, which is solved and has been solved through evolution in different ways, interestingly enough. So that in worms, in C. elegans, both female X chromosomes, there are hermaphrodites in this animal, but it doesn't matter for the story. Both X chromosomes are down-modulated in their expression. so that it equals the male situation. In Drosophila, the single male X is upregulated chromosome-wide in its gene expression to equalize the female situation. And as you may know, in humans and mice, there's a random process of X inactivation. So from cell to cell, either the maternal or paternal X may be inactivated, but the net effect at the tissue level is to equalize gene expression from the X in males and females. But female tissues are therefore mosaic. And I said that historically studies of X inactivation, starting many years ago with the recognition of the bar body as a cytological indication, clumped chromatin against the nuclear envelope in female cells, was useful even for forensic purposes. It goes way back, but some of the themes have proven to be quite universal. So there's, on the X chromosome, transcriptional silencing. There's chromatin condensation, that is the bar body after all. Late replication of the inactive X compared to the active X. DNA methylation of what we call CpG islands. There's CG-rich areas often associated with gene promoters. Specific histone modifications that are inactivating. And expression of a non-translated RNA called XIST, only from the inactive, not the active X. So here's a dramatic cover of a cell from many years ago from Turner's lab showing the inactive X. So the DNA is stained red on all the chromosomes here, and then anti-acetylated H4 is green. And so the inactive X in this chromosome spread is the red chromosome that's different from all of the others. It's just a dramatic demonstration of chromatin modification specific for the inactive X chromosome. That's acetylation of H4 on a particular residue, histone methylation on lysine residues, on lysine tail residues on H3 and H4 are also repressive if they're the correct type of modification. And you can see that here where the inactive X lights up with specific antibodies. And on the bottom panels, you see the non-translated XIST RNA by in situ hybridization, which is bound, it's expressed from and bound to selectively the inactive X. This is just the history of the discovery of this, at that time, unique non-translated RNA, but now we know that non-translated RNAs are associated with epigenetic repression or upregulation in several other systems. And the proof of functionality of the non-translated XIST RNA in the process of X inactivation came, as you may imagine, from mice, where, in this case, Rudy Janish's lab made mice both transgenic for an extra copy of the XIST gene inserted onto an autosome and asked whether the autosome would become partly inactivated, and by his lab and other labs, inactivate the gene in ES cells, in mouse ES cells, and see if X inactivation failed. And so by both of those tests, functionality was demonstrated. And here you just see a kind of nice example where, again, the anti-acetyl H4, which is absent from the inactive X, is in the same place as the presence of the XIST RNA. And this is property conveyed to one of the non-X chromosomes by insertion of the XIST transgene. And lastly, in terms of X inactivation, and again generalizing to other epigenetic phenomena, you have gene silencing actually late in the process of X inactivation by DNA methylation of CpG dinucleotides on cytosine in gene promoters. And this DNA methylation serves to lock in the inactive state. It does so, we believe, not so much by excluding transcription factors that have methylation-dependent binding, but rather by attracting repressive proteins that recognize the densely methylated promoter regions. And among the various epigenetic marks, non-translated RNAs, histone modifications, and DNA methylation, for DNA methylation, this is the type of modification where we understand best how it gets transmitted in somatic cell divisions. And it does this by the DNA methyltransferase class of enzymes, chiefly DNMT1, which has a particularly high affinity for hemimethylated sites in the DNA. So with formation of the daughter strands in S phase, this creates hemimethylated sites, which are quickly recognized by DNMT1 methyltransferase and remethylated, thus propagating the pattern of DNA methylation. And as a corollary of that, if DNA methyltransferases are inhibited, for example, by a drug like decitabine, which is a clinically useful drug for hematologic tumors and now even for solid tumors, methylation patterns can be disrupted. It's called a demethylating drug because it has that effect. And here's another cover of Cell magazine where we have a picture from a paper involving Tim Bester here at Columbia showing that DNMT1 is in the right place at the right time to have this activity. So using anti-DNMT1 antibody, red, and anti-BRDU to show S-phase replication foci, there's a coincidence of the two signals, and giving the yellow color, it's exactly at that location where it's rapidly remethylating those hemimethylated sites. This seminar is not supposed to really get into classical inheritance patterns, but basically the mosaic inactivation of the X chromosome has certain implications for genetic manifestation of genetic diseases, namely the fact that some diseases are X-linked is really nothing to do with epigenetics. It has to do with the single X chromosome in males, and therefore diseases that would otherwise be recessive manifest as X-linked. They show up in males and not in females. But the X-inactivation process does have an implication, and the examples would be Duchenne muscular dystrophy and hemophilia. I'm sorry. It does have an implication, though, for rare unlucky carrier females that can show so-called X-linked, well, they are X-linked, that can show X-linked disorders atypically in a female because of an unlucky pattern of X inactivation such that so you have in the female carrier a mutation on one X chromosome and the other X chromosome is normal. If the X inactivation pattern early in development was unluckily skewed such that the normal X chromosome allele was preferentially inactivated, that X-linked disorder can manifest in that unlucky female. And so that is a consequence of epigenetics. And so that's all I have to say about X inactivation. Again, this study of this important phenomenon provided some of the early data for important mechanisms that are generally applicable in epigenetics. And now let's think about the other famous epigenetic phenomenon, namely genomic imprinting, and consider how this phenomenon has similarities and differences with X inactivation. In X inactivation, as we said, there's a random choice of the allele to be silenced. That is, random choice of which X is going to be silenced in a given cell. In imprinting, it's the parent of origin that dictates the silenced allele. I'll say right now that we know of about 100 imprinted genes. So as I discuss this phenomenon, there's no reason to panic, how important is it or how not important. It affects about 100 genes. And when I say the parent of origin dictates the silenced allele, what I mean is if you tell me the name of an imprinted gene, for example, IGF-2, insulin-like growth factor 2 gene is a classical strongly imprinted gene, I can tell you that your IGF-2 active allele is from your dad, and your silenced IGF-2 allele is from your mom, and it's non-random. It's true for everybody. If you tell me the name of another imprinted gene, such as the H19 gene, which encodes a non-translated RNA, I can tell you that your maternal allele is active and your paternal allele is silent. Another difference, obviously, is that X-inactivation affects the entire chromosome and imprinting is subchromosomal, often involving domains that are on a scale of of several hundred kilobases to a megabase in size. In other words, a little smaller than a chromosome band. X-inactivation is stable in development. Imprinting is often labile, such that, again, if you tell me the name of an imprinted gene, I can often tell you, well, that gene is monoallelically expressed, that is silenced on one copy, only in a part of your brain and not in your blood cells, for example. This is the rule rather than the exception for imprinted genes. Lastly, at the whole tissue level, X-inactivation leaves you with a situation of biallelic expression, because after all, one cell is expressing one of the X's, another cell nearby, maybe the other. So at the whole tissue level, there's biallelic gene expression. In contrast, since, as we said, for a given imprinted gene, your paternal allele is always going to be off, or your maternal allele always off, there's monoallelic gene expression at the whole tissue level. And that has consequences in various situations. So, historically, when did we learn about imprinting? The first hints that this non-Mendelian phenomenon might occur in mammals came from classical embryology in the early 1980s, and I'll show you an example. Later, in the 1980s and early 1990s, imprinted transgenes were described in mice, but it wasn't until 1991 that we knew of, where we could point to a specific gene in humans and mice and say, oh, that's an imprinted gene. And the first examples came from mice, and they were IGF-2, IGF-2R, which is a clearance receptor for IGF-2, and H19, the gene that encodes the non-translated RNA. And actually, our lab was the first to report that H19 was also imprinted in human. And it turns out that IGF-2 also imprinted in human, and IGF-2R, interestingly, not. So, and this brings up the generality that there are a few mouse genes that are imprinted in mice but not in humans, and there are maybe not a lot of genes that are imprinted where you have imprinting in humans and not in mice. Maybe not any, but some would argue with that. Lastly, we now know a lot about the mechanisms of imprinting and the biological rationale for this unusual process. And I've already told you that as we've been beavering away at this, in this field and identifying more and more imprinted genes, the curve of imprinted genes known over the years has become asymptotic to 100. And so I don't think that any additional strong imprinted genes are out there, or maybe just a few are still out there waiting to be found. We reported one new one last year, but that was a lot of work to find that one. Okay. So this is what the embryology looks like. Basically, it was work from these two labs, Serrani's lab and McGrath and Salter, independently making the very similar observations, that if you manipulate mouse embryos, taking out a male pronucleus and putting in a female pronucleus and making a bimaternal, if you will, or gynogenetic embryo, those embryos develop, albeit, and it's shown here, albeit with very rudimentary, relatively rudimentary extraembryonic tissues, relatively stunted placenta and yolk sac, which you see at the bottom of the mouse embryo there. And this was the most advanced mouse embryo that they could get, and it corresponds to about a day 9 stage, which those of you who work with mice, that's not even mid-gestation. Or it is it's about mid-gestation. But after that was no longer viable in the gynogenetic state. The androgenetic conceptus is formed the other way, same idea, having two male pronuclei in the empty egg. Didn't even make it this far, but interestingly, they had a relatively robust outgrowth of the placental tissue. So from these observations, they naturally concluded that it's not enough to have a 2N complement of DNA with a normal DNA sequence to have successful mammalian development. Because after all, these embryos did, and they failed to develop past mid-gestation. You must have half the contribution from mom and half from dad. And this is how they said it at that time without knowing any specific examples of imprinted genes. And now we do. So we mentioned the physical correlates of X-inactivation. Here they are for imprinting. Allele-specific DNA methylation, allele-specific RNA expression, allele-specific histone modifications, localized heterochromatin, replication timing, that's also allele-specific. And the outcome is that imprinting can lead to non-Mendelian inheritance of genetic or epigenetic, as we'll see, disorders. So to make this a little more real, what does allele-specific methylation look like in an experiment? So here you have an old-fashioned southern blot. You can cut the DNA with an enzyme like RSA1 that recognizes an RFLP, a restriction fragment length polymorphism, and distinguish the two alleles that way, and then add to the digest a methylation-dependent, say methylation-sensitive restriction enzyme. Classical one is HEPA2, which will only cut unmethylated DNA. And so for a non-imprinted gene, when you're looking at the promoter region perhaps of that gene, everything cuts. Both alleles are unmethylated. For an imprinted locus, you might see, and do see, this kind of pattern where allele A disappeared and cut down to the short fragments, and allele B is protected because it's methylated. And so this would be an example of a gene that's imprinted and silenced on allele B and active from allele A. An example of such a gene would be H19. Why are some genes imprinted? Really, when this lecture is modified from what I say to the medical students, and the way I put it is, why are any genes not imprinted? Because when you think about the zygote, it forms from fusion of the two cells that are the most different from each other that you could possibly imagine. This huge oocyte and this tiny sperm, where in the sperm head, the DNA is compacted and packaged, and even some of the histones are kicked out and replaced with non-histone packaging proteins. You can't imagine, and methylation completely different between sperm and egg, DNA methylation. So you can't imagine two such different contributions to the zygote, and actually it's a process of active, passive, whatever you want to call it, equalization of those two genomes that leads to genes not being imprinted. And that's what's shown in the diagram here, is that you have this asymmetry between the maternal and paternal contributions initially, and that has to be equalized for almost all of the DNA, except at imprinted loci, where, for example, for H19, paternally imprinted, maternally expressed gene, you have persistence of the hypermethylated state on the paternal copy later in development. So these imprinted genes are genes that escape the normal equalization process that occurs rapidly post-zygotic. And this is really a kind of prettier picture of what I just said, showing imprinting centers that escape this process of genome equalization, epigenetic equalization of the two genomes postzygotically. What does monoallelic expression look like? So here for H19 we've done reverse transcription PCR, and you see in a heterozygous human fetal sample two alleles distinguished by the RFLP with RSA1. But when we look at the RNA, there's only one band, and it's because only one of the two alleles is expressed. And that band, moreover, is reduced in size, and why is that? Because a small intron has been spliced out in a normal fashion from the RNA, and it just shows that we're not getting confused here between DNA and RNA. So really robust monoallelic gene expression. And even more dramatically, when one makes a knockout mouse targeting an imprinted locus, you can get this type of effect. And this is a classical old-fashioned northern blot showing expression of another imprinted gene, FILDA2, which, like H19, is maternally expressed, paternally silent, in mouse embryos with different genotypes. You see on the left the wild type ++ genotype, no deletion. Then you have a heterozygote where the maternal allele's been knocked out and paternal allele's still there, no expression. And then the opposite heterozygote, same DNA situation, but just a different parent contributing the knockout allele, and you have normal expression. Another wild type ++, and then a complete knockout. Of course, there's no expression. So the maternal heterozygote and the paternal heterozygote are totally different. Imprinting is, when it occurs, is a very potent mode of gene regulation. It can turn off the gene on one allele. For those of you who are interested in human genetics, uniparental disomies, which occur when the parents carry chromosome translocations, like particularly Robertsonian translocations in the offspring, you can get uniparental disomies, which are illustrated here, in which there are, for example, 2 copies of chromosome 12 material in the offspring, so that you have a normal amount of chromosome 12, but both copies came from mom. And when you have that kind of UPD12 situation, you can ask, is anything wrong with that person? Is anything wrong with that mouse where only chromosome 12 was inherited from only one parent, but in the 2n amount? And some UPDs have a phenotype, and other UPDs don't. Other UPDs are normal. And so by mapping which UPDs lead to an abnormal phenotype, it was possible to say this chromosome region in mouse, this chromosome region in human must contain at least one phenotypically relevant imprinted gene. The other types of mapping were by chromosome walking, and now, of course, whole genome approaches. Now we start to get at the question of why imprinting exists. I mean, in other words, why has this unusual phenomenon persisted, and with almost perfect conservation, in fact, between human and mice? This is a question for evolution. It's an evolutionary biology question, which can be addressed, as I'll show you, partly in the lab. And the first question is, what types of phenotypes does one observe in knockouts or UPDs or even transgenic addition of imprinted genes? And the phenotypes turn out to be all one of 2 types. Growth, either overgrowth or undergrowth phenotypes, or behavioral phenotypes, either hypo or hyperactivity of some sort. Under the category of growth, I'll also include metabolic effects. And at a very early point, as data were starting to come in, really only really in the same year as the first papers appeared identifying imprinted genes in mice, David Haig and his collaborators proposed this conflict model to explain the evolutionary conservation of the phenomenon of genomic imprinting. And I'm discussing this model because I believe it's correct. And the reason I believe it's correct is as we've identified more imprinted genes, they proposed this model, in fact, based on only the first two examples, IGF2R and IGF2. But now, based on all of the examples that we know, the predictions of this model have either been verified, validated, or the example is uninformative with regard to the model. So no failures. And the idea is that maternally expressed imprinted genes often retard growth of the conceptus and or constrain demands on maternal resources, which I'll have a little more to say about, whereas paternally expressed genes often have the opposite effect, promoting growth and leading to increased demand on maternal resources. And here we have the human chromosome ideograms with the best studied imprinted genes, which I've color-coded to indicate whether they affect growth, behavior, or both. And nearly all of them can now be color-coded by these processes, which is consistent with the Haig hypothesis. And moreover, the direction of all of the ones that I've color-coded is consistent with the predictions of that model. Now, why would this be? Basically, the idea is that in mammals, the paternal alleles and maternal alleles have different, say, strategies that will allow them to propagate successfully in the population, and that's what evolution is all about. And since mom devotes an inordinate amount of resources to each conceptus, whereas dad devotes his sperm, basically, the strategies are different, so that a paternal allele will spread in the population more effectively if expression of genes from the paternal copy promotes conceptus growth and demands on the mother, whereas the mother will spread her alleles in the population more effectively if she can reproduce multiple times over her lifetime. And in order to do that, has to constrain in an optimal range the demand, the size of each conceptus and the demand of each conceptus on her resources. It turns out, I'll show you an example of imprinting in the placenta, which is very relevant to this idea, and also where imprinting has been found in marsupials. It's been found exactly according to the predictions of this model in the lactation, in the gland in the pouch. Remember, in marsupials, it's not fully developed, the offspring's not fully developed, and stays in the pouch and suckles for a long period of time. So the demand on mom's resources, rather than being from the placenta, through the placenta, is through the mammary gland, if you will. It must not be called a mammary gland, but whatever it's called, in the pouch. The nipple is actually there in the pouch, and that tissue has strong imprinting of imprinted genes in marsupials. And that research is ongoing, mostly in Australia, as you might imagine, and is very interesting. So, but here's the example from our lab a number of years ago where we identified this FILDA2 gene, which is maternally expressed, paternally silenced imprinted gene. And I'm showing you here just a diagram of the conceptus, and you have the yolk sac, the labyrinthine layer of the placenta, which corresponds to the free chorionic villi in humans. And this is where nutrient exchange occurs, early and late in development. And this is exactly where here we just have antibody staining of FILDA2 protein. This is exactly where the protein is most highly expressed, in the labyrinth of the placenta and in the visceral endoderm of the yolk sac. So it's expressed in tissues that mediate maternal-fetal exchange. And what happened when we knocked out this gene, either knocking out only the maternal allele or both alleles? At first, we were staring at these mice. Dale Frank was doing a postdoc in my lab at the time, and he made these mice, and everything looked kind of normal. And you can see here the histology of the placenta looks normal. And I was getting frustrated, and then Dale said, well, Ben, if you're frustrated, why don't you just weigh the placentas? And it was the weights, which was Dale's. I mean, it was his mice. But anyway, so the weights of the placentas, that's the only phenotype. And it matches exactly the Hague hypothesis prediction. You see overgrowth. Well, usually, if you delete a gene from a mouse, you might see some detrimental effect. Here, we deleted the gene, and the placenta got larger. And it had no benefit to the fetus. The only effect was to drain, if you will, maternal resources. It's an overgrown placenta that has no benefit to the fetus. So exactly consistent with the Hague model. It turns out, interestingly, we want to talk about medicine, that the FILDA2 gene has a polymorphism in its promoter region in humans, and that polymorphism is genetically associated with low birth weight or lower birth weight. So if you have the high expressed allele, you have a tendency to have a lower birth weight. And that corresponds to transgenic data where we've added extra FILDA2 in contrast to the knockout. And when the placentas are smaller due to the extra FILDA2, that is detrimental to the fetus, to the growth of the fetus. So let's talk a little bit about growth syndromes in humans due to problems with imprinted genes. And then I want to end with a sort of extra credit topic that's very relevant, I think, to genomic medicine and complex diseases, common complex diseases, namely the effects of human haplotypes on DNA methylation patterns. So just to consider first the overgrowth and undergrowth disorders in humans that are due to problems with imprinted genes. The overgrowth disorder is called Beckwith-Wiedemann Syndrome, and it leads to a very dramatic in utero overgrowth and then a large baby. And the opposite, if you will, phenotype syndrome is called Silver-Russell syndrome, which leads to a rather dramatic growth failure that is severe intrauterine growth restriction, or IUGR. Beckwith-Wiedemann syndrome, the overgrowth syndrome, is associated with an increase in childhood cancers, notably Wilms tumor and some others. And we'll have something to say about that. In terms of the other aspect of biology that's controlled by imprinted genes, neurobehavioral traits, Prader-Willi syndrome is the famous example where you have behavioral and other problems. Initially, PWS individuals are floppy babies that make low demand on maternal resources in the sense that they don't suckle. But later in life, they have problems with obesity because of inability to regulate appetite. So it's an interesting disorder, again, consistent with the Hague predictions. And Angelman syndrome is a severe mental retardation syndrome, both, again, caused by problems with imprinted genes. So what are the types of molecular lesions that affect these genes in human disease? We've already mentioned uniparental disomy. You can see, not infrequently, particularly for PWS and AS, deletions of imprinted chromosomal regions. One can see a little less frequently point mutations in imprinted genes, and then a pattern of inheritance that is non-Mendelian. And then some of the more interesting problems, microdeletions of imprinting centers. These are small kilobase size DNA sequences that regulate imprinting over the larger domain. And so some examples of imprinting disorders can be due to DNA microdeletions of these particularly important sequences. And then, of course, the most interesting situation is where one has an epimutation, where there's no change in the DNA, but there is a change in DNA methylation, which occurred presumably in a stochastic fashion early in development and leads to the full-fledged syndrome. And this is a true non-genetic, epigenetic cause of human disease. And this really happens, as I'll show you. And then lastly, loss of heterozygosity in cancer, particularly in pediatric solid tumors, can span imprinted domains and lead to the sort of growth phenotype that's associated with embryonal tumors. So in Beckwith-Wiedemann syndrome, this is the phenotype, X omphalos, which is partly presumed to be overgrowth of the internal organs and partly failure of abdominal wall closure. Macroglossia, very characteristic overgrowth of the tongue, and gigantism. Ear pits and creases are incidental diagnostic findings, helpful findings. Organomegaly I referred to already. And then at the histological level, I'll show you some pathology. Hemihypertrophy in some cases, neoplasia in about 7% of cases, and we can now do DNA diagnosis for which of those cases are going to be at highest risk, again, as I'll show you. And normal intelligence and lifespan, and even the face structure becomes more normal looking with age as the jaw grows to accommodate the larger tongue. So here's an example of a patient that was seen here with Beckwith-Wiedemann syndrome, just a huge baby where you can barely make out there's a scar, abdominal scar from an omphalocele repair, and it's easy to see the macroglossia. The kidney, not in this baby, but in other cases of Beckwith-Wiedemann syndrome, particularly those predisposed to Wilms tumor, which is a pediatric kidney cancer, will have nephrogenic rests, which are primitive cells that persist in the renal cortex. They shouldn't be there after a certain point, and yet they persist in cases of Beckwith-Wiedemann syndrome that are predisposed to subsequent Wilms tumor formation. But this can be massive overgrowth. These are the embryonal tumors, and the loss of heterozygosity that occurs in these tumors, which gave a clue, actually, to the Beckwith-Wiedemann syndrome locus early on, is on chromosome 11p15, and that is where the BWS syndrome turns out to map. So these are the causes. In band 11p15, you have three relevant genes, CDKN1C, which encodes a cyclin CDK inhibitor and is imprinted, and H19 and IGF2, which are also imprinted. About 10% of cases, maybe a little less, of Beckwith-Wiedemann syndrome are due to mutations in CDKN1C. It's maternally expressed. It's a growth inhibitor. So I could have told you it's maternally expressed, paternally silenced. And it is, I don't know, it makes sense. There's uniparental disomy cases, there's chromosome translocations, but most cases are due to epimutations. So not only are epimutations real, but epimutation accounts for most cases of Beckwith-Wiedemann syndrome, with mutations accounting for only a small minority. And these epimutations occur in one of two imprinting control elements, both in the same chromosome region, which are shown here. Sometimes they're called ICR1 and ICR2. One of them is upstream of H19, and the other is near the CDKN1C gene. It turns out that when the epimutation occurs in this region near CDKN1C, omphalocele is very severe and common, and Wilms tumor very rare. But when the epimutation occurs upstream of H19 in that ICR1, Wilms tumor is common, and omphalocele also occurs sometimes. So a DNA diagnostic test that it can be very useful and is performed routinely now is to ask which of these two regions has the abnormal methylation in this child with Beckwith-Wiedemann syndrome. And if it's the ICR1 region, it makes sense to ultrasound that child more frequently or other types of imaging to see if there's tumorigenesis. If it's in the other region, one can relax a little bit about this. And so there are actual clinical protocols based on this idea, DNA diagnosis. And here you see one of Wendy Chung's patients where we did a southern blot diagnosing that the problem here was loss of methylation in the region near CDKN1C, ICR2, and therefore was reassuring to the family that this would not be a high risk for cancer. And indeed, cancer has not arisen in this individual. The opposite, Silver-Russell syndrome, phenotypically opposite, but mapping to the same chromosome region in a lot of cases. And you also see this asymmetric growth, just like in Beckwith-Wiedemann syndrome, you can have hemihypertrophy. There's asymmetric growth restriction in Silver-Russell syndrome. It's just a clinical observation. I don't know mechanistically what this really is. Presumably there's some tissue mosaic effect. But as this syndrome was mapped and candidate genes were examined for DNA methylation abnormalities, principally by Christine Jiquel's lab in Europe, and now I think she's moved to Australia, the syndrome mapped to IGF2H19. Exactly the same locus as Beckwith-Wiedemann syndrome, exactly the same DMR, the ICR1, but the opposite change in methylation. So in Beckwith-Wiedemann syndrome, one sees gains of methylation in this region, and in Silver-Russell syndrome, losses. So how can we bring all of this together in a mechanistic way? It's worth looking at this, I think. The normal situation for H19 and IGF2 is that they're separated, if you will, by this ICR1 DMR, which is a CG-rich region that binds a protein called CTCF. And that protein, CTCF, is responsible for creating an insulator element. It binds at the base of chromatin loops. And its binding, in this case and in many other loci, is methylation dependent, such that the chromatin loop only forms, CTCF can only bind, and the loop can only form when its binding site is unmethylated. And when its binding site is methylated, it can't bind, and the loop doesn't form. So, in the normal situation where H19 is expressed from the maternal copy, CTCF is bound there, and that region is unmethylated, while on the paternal copy, that region is methylated, CTCF is not bound, and the IGF2 promoter can interact with a downstream enhancer because it's not blocked by the CTCF binding. That's normal. So you have monoallelic expression of both genes, but from opposite alleles. In Beckwith-Wiedemann syndrome and Wilms tumor, where there's overgrowth, both alleles become methylated. There's no binding of CTCF. IGF2 is active now from both alleles, doubling its dose. It's a growth promoter. And H19 RNA, which can be growth suppressive, particularly interestingly in the placenta, is turned off. And the net result is overgrowth. Opposite in Silver-Russell syndrome, where you have loss of methylation on both copies, there's lack of methylation on both copies, CTCF bound to both copies, overexpression of H19 and no expression of IGF2. And this leads to the severe undergrowth. So a quite remarkable story that evolved over 20 or so years, but in the end, very satisfying. The other famous diseases I mentioned, and now just in 5 minutes, I want to go over this last issue of interactions between genetics and epigenetics. These interactions can be either in cis or trans. And I think cis and trans interactions of the genome with the epigenome is a very important post-genomic, so we're in the post-genomic era now. This is a very important post-genomic topic to think about. In terms of trans interactions, an increasing, I actually submitted this chapter for the course reading that we wrote for the Emery and Rimoyne, standard Canadian genetics textbook, and you see the number of diseases that are linked now to mutations in genes that encode epigenetic modifiers, such as DNMTs, histone modifying enzymes, and so on, has just mushroomed. And so and these mutations in genes that read and write epigenetic marks are starting to explain phenotypes that we would previously have thought of as non-genetic, even, like many cases, it turns out, of congenital heart disease, that in past years people have sought environmental explanations for these isolated non-syndromic cardiac defects, which we now know in many cases can be attributed to mutations in epigenetic modifier genes. And the names of those genes and the enzymes they encode and the histones that are acted on are just in this diagram from the nice paper from Rick Lifton's lab, which has been followed up by Wendy Chung and Dorothy and people here with larger studies confirming this idea of a relatively high frequency of mutations in this particular type of gene, the epigenetic readers and writers in congenital heart disease. Just one example. And then something that we've worked on and published on starting in 2008, which is haplotype-dependent DNA methylation patterns. I'll show you what this looks like. So here we used old-fashioned SNP chips to score allele-specific methylation, and you see the sort of primary data on the left. One allele lights up, and we added, as a pre-digestion of the genomic DNA, a methylation-sensitive restriction enzyme, one of the alleles still lights up and the other allele drops out. And this is an indication of allele-specific methylation, but not at an imprinted gene. It's a gene that's non-imprinted, cytochrome P450-2A7. But the allelic asymmetry in methylation is just as strong as one would see at an imprinted gene. It turned out that, and it affects gene expression, it turned out that for many of these loci, we could predict which allele was methylated and which unmethylated, not by parent of origin, but rather by the genotype at adjacent SNPs. In other words, it's the haplotype that is determining this kind of allele-specific methylation. And we call this hap-ASM, haplotype-dependent allele-specific methylation. It's a very strong correlation. And this is the context. So you have the same outcome in the cell, basically, but for two different reasons. At an imprinted locus, this would be a parent of origin dependent effect. At these hap-ASM loci that are more common, actually, than imprinted loci, there's about 1,000 of them, you have the same outcome, but it's dependent on the local haplotype acting in cis to establish the DNA methylation pattern. It has a practical application that's now being pursued by us and very aggressively by quite a number of other labs. And that has to do with extracting information from genome-wide association studies. So as many of you know, GWASs have been successful in some diseases and less in others at finding disease genes that usually have modest odds ratios if you tell me this is a risk allele, but reproducible. The problem with GWASs is that nearly all of these hits greater than 90% of them are not changing amino acids in coding sequences. Rather, they're affecting presumed regulatory elements. So one doesn't really have, in a pure GWAS, anything to go on beyond the statistic. There's no biology there to say that this really is an important regulatory element. Secondly, because of the Bonferroni correction problem in GWASs, there are a lot of interesting sub-threshold peaks. You have to have a p-value of 10 to the minus eighth to accept a statistical peak in GWAS as being valid. But there are many interesting signals at 10 to the minus 6 p-value that might be biologically true and interesting and point to disease pathways. So how can one get over these roadblocks? Allele-specific methylation mapping is very helpful. And this is just from a review article that we published a couple of years ago, where the strategy is to take these sub-threshold and super-threshold and even, quote, non-replicating GWAS peaks and overlap that set of loci with genes that show allele-specific methylation or comparable phenomenon like allele-specific expression that's haplotype-dependent. And in that overlap, you must be seeing true biological positives because you have physical evidence that the two haplotypes are different. So this must be a true regulatory sequence. You have more methylation on one than the other in person after person after person in a reproducible fashion. So this type of mapping is ongoing. And in fact, I'll just end by saying that as we've mapped, and this is very interesting, the DMRs for this hap-ASM phenomenon, and here you see a heat map of the allelic asymmetry for one locus, and you can see bisulfite sequencing on the bottom indicating that the allelic asymmetry is only seen in regions G and a little bit in region F there. There's a very discrete DMR which precisely overlaps, and now I really should point, I guess, but it precisely overlaps with an ENCODE chromatin track showing the CTCF binding site. And then just another example, exactly the same finding. The strong allele-specific methylation, and this is haplotype-dependent, not imprinted, again, precisely overlaps the CTCF site. So that not only imprinting, but also haplotype-dependent allele-specific methylation often, though not always, maps to CTCF sites. So the mechanism may be that when you have a polymorphism in the site or near the site that is part of the haplotype, the CTCF can no longer bind, and methylation creeps into that region, leading to the allele-specific methylation in a heterozygote. So one haplotype is a good CTCF binder, the other haplotype not, and methylation asymmetry is established based on that. In fact, we've now done enough whole genome analysis of hap-ASM loci that we can test this idea, and it turns out to be true. There's a strong enrichment for polymorphic CTCF motifs, but not invariant CTCF motifs in hap-ASM loci. And this is I know, very technical, but it's the last thing. The conclusion is that variable human traits and disease susceptibility can be due to individual differences in chromatin looping. And this phenomenon is in turn haplotype linked and DNA methylation dependent. It's really, for those of us who think about epigenetics, a very satisfying outcome because it so nicely ties together genetics, human genetics and epigenetics. And to show it schematically, basically, part of the reason that you and I look different, the person sitting next to you, is you have different chromatin loops. And some of those chromatin loops are different in a DNA methylation dependent way, which is also depending on your haplotypes.